In this section, we'll talk about the chemical nature of neural communication. We'll talk about the different chemicals in the brain and how they affect neural function. Neural transmission or chemical transmission relies on a stereotypical set of processes that are critically involved in passing signals between neurons. Neurotransmitters have to be synthesized, and as and when an action potential reaches the presynaptic cell, they have to be released by the synaptic vesicles that are storing them into the synaptic cleft. These neurotransmitter molecules will then either bind to receptors and result in depolarization in the postsynaptic cell, or perhaps even hyperpolarization, which is the inhibitory signal. It depends on which ions are let in or out. And much of the neurotransmitters that are released into the synaptic cleft are then either inactivated or diffuse away, or are actively taken back up into the presynaptic cells or the glial cells in that area, or degraded through enzymatic action. So inactivation of neurotransmitters is a critical step in the life cycle of a neurotransmitter. This finely controlled process of synthesizing, storing, releasing, inactivating, and specific effects in the postsynaptic neurons are very critical to the function of a particular neural circuit. This is what is happening at every synapse all of the time. Once again, notice that depending on whether the ion channels that are affected in the postsynaptic cells let positively charged ions in or out, or negatively charged ions in or out, or differentially allow more positivity or more negativity inside, we can end up with different scenarios of excitation and inhibition. Depolarization in the postsynaptic cell leads to excitatory postsynaptic potentials, and hyperpolarization in the postsynaptic cell leads to inhibitory postsynaptic potentials. The neurons then, in their role as postsynaptic cells, are busy integrating information from multiple inputs, some of which may be excitatory and others which may be inhibitory. And the activity in that neuron and the signal it sends down its axon then depends on the net positivity or negativity that is generated in the cell body as a result of all these inputs at any given point of time. In this way, the depolarization that collects within the cell body can affect ion channels at the axon hillock, which is the start of the axon. All these voltage sensitive channels slowly start opening as the depolarization builds. At threshold, a spike is generated. And this is how neural integration results in activity in a postsynaptic cell. The neurons then accumulate inputs both over time and over space, by which I mean all the synapses on all of the dendrites, and produce an output that is a function of its inputs. In the top panel here, we see a neuron at rest with multiple synapses that are inactive. In the second panel, as an excitatory fiber is stimulated, an action potential travels down that fiber and results in a small amount of depolarization in the postsynaptic cell. If at the same time another fiber was active and the resultant depolarization was enough for this cell's threshold to be reached, an action potential is fired in that cell as is indicated by the axon turning red. If, however, at the same time, inhibitory input is also generated, then you can see that there is a mathematical kind of summation of depolarization and hyperpolarization within that cell. The graphs actually show very interestingly both the summation of the positivities and the negativities, but also the time lags between the different activations. So the profile of activity within a cell then is a sum of all of its inputs over time and space for all of the active synapses. This fine-tuned ability to modulate the response of a postsynaptic cell through multiple inputs results in what we call integration of inputs or neural processing. In this way, the firing rate of a cell increases as the excitatory inputs increase. So an increase in a stimulus, for example, will increase the number of action potentials fired by the sensory neuron or the cell that responds to that stimulus. This, in a sense, is how the neurons code for information. The firing rate of action potentials is the message that a neuron communicates to other neurons within the circuit. Since much of what happens at the synapse relies on the nature of the neurotransmitter that is released and taken up by the postsynaptic cell, the neurotransmitters play a critical role in generating excitation or inhibition within the system. There are many kinds of neurotransmitters. 9 of them are particularly important to understanding how we think, feel, and behave. So we'll talk briefly about those, but it's important to realize that there are many neurotransmitters within the central nervous system and the peripheral nervous system, some perhaps as yet undiscovered. Acetylcholine is a neurotransmitter which is found largely in the cerebral cortex and is critically involved in motor control in the muscles and in learning, memory, sleep, and dreaming in the central nervous system. This is a neurotransmitter which is involved in Alzheimer's, for example. It's also the target of the mushroom hallucinogens. Epinephrine and norepinephrine regulate energy levels and levels of arousal and attentiveness. These also play a role in initiation of hunger, attention, and circadian rhythms. Epinephrine and norepinephrine are often the target of stimulant drugs. Serotonin is a very important neurotransmitter in the brain, found all across the cortex. It is heavily involved in control of behaviors, impulsivity, aggression, depression, food intake to some extent, sleep-wake cycles, modulation of brain movement, breathing, and social behaviors and emotions. Serotonin belongs to the same family of neurotransmitters as dopamine. Dopamine is found in the basal ganglia and is related to a network that controls muscle rigidity and another that is very critically involved in reward and pleasure. It is then the neurotransmitter which is often related to psychotic behaviors. GABA is found in most regions of the brain. It is the primary inhibitory neurotransmitter in the central nervous system and is the target of most depressant drugs. GABA is largely responsible for anxiety reduction within the brain. Glutamate, conversely, is an excitatory neurotransmitter, also found in almost all parts of the central nervous system, and it enhances action potential, is therefore very critically involved in learning and memory. Other neurotransmitters like endorphin and substance B are related to pain perception. Endorphins are also critically involved in reward pathways and pleasure centers. They are opioid-like chemicals that occur naturally in the brain. Because each type of neurotransmitter is so critically involved in different types of behaviors, any drug that affects either the synthesis or the storage or the release or the degradation or the reuptake or the binding of these neurotransmitters in the synapse can affect behavior in very profound ways. Drugs that enhance neurotransmitter action are called agonists, and those that inhibit the action of neurotransmitters are referred to as antagonists from the word antagonize. And in this way, many chemicals can in fact influence how we think and feel. Other chemicals that are critically involved in changing brain activity are known as neuromodulators. These alter how neurons exchange messages. Some neurons release these substances into broad areas and affect the activation or the level of activity in many neurons simultaneously, hence the term modulation. For example, acetylcholine does that in widespread areas of the brain. So do the opioids. These change the conditions for neural firing and might in fact, for example, have an alerting role or an arousal role in the brain. Drugs, on the other hand, seem to have specific effects on circuits and affect neurotransmitter release or neural firing in a more specific way. For example, morphine binds to opioid receptors and enhances the activation of those circuits. Many psychoactive drugs that are often used for recreational purposes that you may be familiar with, hopefully you're not familiar with most of them, work through changing neurotransmitter activity. So on this slide then, I have for you some examples of common psychoactive substances and their effects on the neurotransmitter systems. Alcohol, for example, is a depressant and acts primarily through its effect on the GABA circuit. Amphetamines affect dopamine and norepinephrine. Caffeine is an adenosine agonist. I didn't talk about adenosine earlier. If you're interested, you can look that up. One that I like to talk a little bit about is the GABA agonist rohypnol, which is also often referred to in colloquial terms as a roofie. This affects the GABA circuits to the extent of extreme depression, where the person's arousal levels then are very, very low at that point. Notice it's an agonist, so it enhances the action of GABA in the brain. I hope this helped you to appreciate the extent to which the chemical signals in the brain regulate our levels of different behaviors and how any change in those chemicals or the neural activity relying on these chemicals can then result in profound changes in behaviors, thinking, decision making, or even energy levels.